In this video, we are going to be covering the topics in Chapter 19 and 20, which have to do with the citric acid cycle and oxidative phosphorylation. So before we talk about the process of the citric acid cycle and oxidative phosphorylation, we must establish that these pathways are going to be connected to something that we talked about in Chapter 17, which is glycolysis. Understand that in the process of glycolysis specifically, which we introduced in Chapter 17, that talks about the conversion of one molecule of glucose to 2 molecules of pyruvate. In this figure, you don't see the 2 molecules of pyruvate because it's talking about the molecules that are made in general. But understand that what we are going to be introducing in Chapter 19 and 20 is how that pyruvate is going to be creating new molecules that are going to be resulting in the synthesis of the energy currency of the cell, which is ATP. Remember that ATP is going to be a nucleotide. And remember that is adenosine triphosphate. So when a metabolic pathway breaks down nutrients, understand that that is known as catabolism. So the process of glycolysis, in which now glucose is going to be turned into pyruvate, that's an example of a catabolic pathway. Specifically here, we're going to continue to break down that nutrient. So all of these processes are catabolic. Now, in the last chapter, in Chapter 18, we introduced gluconeogenesis. Gluconeogenesis was the process by which we have different molecules, but ultimately the one that we described specifically was the process of pyruvate being converted all the way back to glucose. That's an example of an anabolic pathway or anabolism, because remember that through that process, that's going to be the reductive synthesis of biomolecules. So we're going from small number of atoms into making larger biomolecules. Now, understand that organisms are going to obtain more energy from nutrients by aerobic oxidation than anaerobic oxidation. So these processes that we're going to be discussing in Chapter 19 and 20 is under aerobic conditions. And remember that aerobic conditions means that we are going to have molecular oxygen available. Now, we have so far discussed in Chapter 17 that the process of glycolysis has a net of 2 molecules of ATP made from 1 molecule of glucose. But in Chapter 19 and 20, we are going to be exploring how, once that pyruvate is made and through the aerobic process of the citric acid cycle and then oxidative phosphorylation, we can make anywhere between 30 to 32 molecules of ATP, okay? And all of those are coming from 1 glucose molecules. Let's also think about that glucose is not the only biomolecule that is going to be producing energy in the cell. Fatty acids, which are going to come from your triglycerides, and even amino acids that are going to come from the protein that you consume, okay, can also be processed through these pathways, and they produce energy. But we are not going to be focusing on fatty acids and amino acids metabolism at this moment. Now let's go over the overall view of the citric acid cycle. So understand that in eukaryotes, the process of glycolysis and the citric acid cycle, which is our focus in Chapter 19, is actually compartmentalized. The process of glycolysis actually occurs in the cytoplasm, while the citric acid cycle occurs in the mitochondria. So first we must establish, because the processes that we're going to be talking about in chapter 19, it is in the mitochondria. What are the components of the mitochondria? So as you can see here, mitochondria is composed of an outer membrane, which is going to be the outermost membrane. It's going to have an inner membrane, which is going to be this line that I'm going to be drawing around, and I'm partially just drawing it. So that's the inner membrane of the mitochondria. It's going to have a cristae, and cristae is going to be the crests that are present in the inner membrane. And then the space inside the inner membrane of the mitochondria, that's what is known as the matrix. Understand that most of the enzymes that are part of the citric acid cycle are actually going to be present in the mitochondria matrix. Now, as you can see on the right side of our slide, here we have the actual cycle, okay? So understand that the first step of the cycle is going to be when the molecule of acetyl-CoA reacts with oxaloacetate to make citrate. Okay, because we're forming citrate, this is what gives the name for the pathway. Now, at times you will hear me say TCA cycle, which is a tricarboxylic acid cycle, or you will hear me say Krebs cycle. All of those three names, citric acid cycle, TCA cycle, and Krebs cycle, all are names utilized for this pathway. Now, one of the things is that this is called a cycle because, as you can see in the first reaction, as I just described, and we're going to talk about in detail, we are combining acetyl-CoA and oxaloacetate. As this molecule that produces citrates is cycled through the whole process, we are also going to be producing overall oxaloacetate. So that's why it is called a cycle, because we're starting with combining the product of the cycle with the first incoming molecule. So understand that the citric acid cycle is going to be composed of 8 steps, and we are going to talk about them in detail in terms of what is the reaction and what is the enzyme. When it comes to the enzymes, understand that for the exam, you need to know the classification of each of these enzymes. Now, let's talk about the cycle itself in detail. So before we talk about the 8th step, the pyruvate molecule needs to be prepared to enter the cycle. So before we even talk about the 8 steps, understand that there is a preparatory step, which means that pyruvate is going to be converted into acetyl-CoA. Acetyl-CoA is the molecule that enters the TCA cycle. So pyruvate directly doesn't enter. It needs to be converted into acetyl-CoA before we even get started. So how is that accomplished? Well, pyruvate, as you can see, this is going to be a 3-carbon molecule. Is going to be converted into 2 carbons. So how do we convert that? Through the process of oxidative decarboxylation. The enzyme that catalyzes this reaction is called pyruvate dehydrogenase. But understand that we know it as a complex because understand that overall, this pyruvate dehydrogenase is going to be composed of 3 enzymes and 2 cofactors. So as you can see here, we have FAD, which is a nucleotide derived molecule that is involved in reaction. We have, and this is a coenzyme. We have also NAD, okay, that is going to, as you can see, be reduced to NADH. We have coenzyme A, which is another molecule that it is a nucleotide. And as you can see, we have TPP, which this is known as thiamine pyrophosphate. We have lipoic acid. We even have a divalent cation in this one specifically is magnesium. So overall, what is occurring in this oxidative decarboxylation? As you can see, and I'm going to be circling in different colors. The reason why this is a decarboxylation is because we're going to be removing that carbon and 2 oxygens in the form of CO2. Then, understand that specifically coenzyme A is going to be providing this S-CoA into the acetyl group, which is this. That's why the molecule is known as Acetyl-CoA. One more thing that I should establish is that this is a functional group that we haven't talked about because it's very particular to this system. This is known as a thiol ester bond. And we are going to be looking at its importance later in these videos. Now understand that the whole process of converting pyruvate to acetyl-CoA is an exergonic process, which means that energy is going to be released overall when this reaction happens. Now that we have synthesized acetyl-CoA, we are going to start talking about how acetyl-CoA enters the pathway and it is overall cycled through the citric acid cycle to generate oxaloacetate. So, before we get started, one more thing that I have to mention before we dive into the citric acid cycle. Remember that who's contributing the S-CoA portion to the acetyl group that came from pyruvate is going to be coenzyme A. And coenzyme A, we talked about it previously. Now, coenzyme A has 3 different components. Coenzyme A has an amino ethane thiol. The next component in coenzyme A is pantothenic acid. And then lastly, as you can see here, it has a 3' phosphorylated ADP. That's why coenzyme A is known to be a nucleotide, because as you can see, the last portion of this molecule. And again, this is the molecule that gets incorporated into the acetyl group that came from pyruvate, okay? And the molecule of acetyl-CoA is what enters the TCA cycle. Now, we are going to talk about the steps in the TCA cycle. So, step 1, formation of citrate, okay? So when I discuss the TCA cycle or the citric acid cycle, and for those of you that are like, that couldn't catch the names, the citric acid cycle is also known as TCA cycle because of the tricarboxylic acid that is made actually in step 1. It is also known as Krebs cycle. So whenever you hear citric acid cycle, TCA cycle, or Krebs cycle, understand that all of them mean the same pathway. So as I was mentioning, on step 1, we're going to have the formation of citrate. So oxaloacetate, which is a 4-carbon system, is going to be reacting with Acetyl-CoA, that is a 2-carbon system, to make a 6-carbon molecule. As you can see, this condensation reaction, it is catalyzed by citrate synthase. And citrate synthase, as you can see, is taking these 2 molecules and gluing them. But as you can see, we are not hydrolyzing ATP. So this gives you the clue that citrate synthase is a lyase. The process of this condensation reaction is actually an exergonic process. So as you can see, energy is released when this reaction happens. Now, let's talk about the second reaction that occurs in the system. So when we have citrate, then citrate needs to be isomerized to isocitrate, okay? Because if we look at the molecule of citrate, this is going to be a tertiary alcohol, okay? When isomerization happens, this forms a secondary alcohol. Okay, so aconitase is going to be the enzyme that catalyzes this reaction. Now, some of you will be like, easy. We are isomerizing from a tertiary alcohol to a secondary alcohol. So aconitase must be an isomerase. That is incorrect. Aconitase is actually a lyase because this isomerization from tertiary alcohol to secondary alcohol actually undergoes an elimination addition reaction that can be outlined in the bottom of the molecule. As you can see, citrate gets dehydrated. It forms a carbon-carbon double bond. So after the dehydration, we're going to be making an alkene. Then that alkene, we're going to be doing a hydration reaction. So this is dehydration followed by hydration. In a way, elimination followed by addition. And that's how the molecule results in the formation of isocitrate. So remember, when it comes to this, even though overall is an isomerization, this is not an aconitase is not an isomerase reaction, or it's not an isomerase enzyme, I should say. Understand that aconitase needs a divalent cation, in this case iron, in order to have this reaction accomplished. This process is actually endergonic, and you may be asking yourself, okay, so if this process is endergonic, how can it even proceed? Remember the first step that we just discussed, when acetyl-CoA binds or reacts with oxaloacetate, is an exergonic process. So the energy in order for this reaction to occur actually comes from the energy that was released on reaction number 1. So on step 3 is the formation of alpha-ketoglutarate, okay? And when it comes to alpha-ketoglutarate, understand that we are going from isocitrate, okay, which is a secondary alcohol. And as you can see here, the keto portion of alpha-ketoglutarate comes from the formation of this ketone. In the process, we're also releasing CO2. So that's why the reaction to convert isocitrate to alpha-ketoglutarate is known as oxidative decarboxylation. Now, one of the things that I want to outline, why is it that in step 2, we actually had to isomerize a tertiary alcohol to a secondary alcohol? Remember from your organic chemistry, tertiary alcohols are not reactive when it comes to oxidation. they cannot be oxidized, only primary and secondary. So in this cycle, makes sense that on step 2, since the tertiary alcohol cannot be oxidized to any functional group, it needs to be converted to a secondary alcohol, because as you can see in the next step, on step 3, we're forming a ketones. Ketones cannot come from tertiary alcohols. They can only come from secondary. So that's the logistics of why we did the isomerization on step 2. So the process of converting the isocitrate to alpha-ketoglutarate, as you can see, is catalyzed by the enzyme as isocitrate dehydrogenase. So overall, as you can see, first, what happens in the process is that we are going to be oxidizing our isocitrate to making this molecule that is called oxalosuccinate. And then oxalosuccinate gets decarboxylated, meaning that's where the CO2 comes from, to give rise to alpha-ketoglutarate. Now, this process of the oxidative decarboxylation of isocitrate, as you can see, is an exergonic process, so it's going to release energy.